Dear students, after studying this module, you shall be able to know about enzymes functioning as drug targets, learn about enzyme structure and catalysis, also know about the concept of enzyme inhibition, and also understand different types of enzyme inhibition processes. So, let us begin this topic with the introduction to the fact or to the topic of why enzymes as drug targets. What is the need? Medicine in the 21st century has become a science in which drug molecules are directed against the macromolecules. Enzymes hold a prominent position amongst the biological macromolecules that can be used as good drug targets. Some features of enzyme structures and reaction pathways which make them very attractive and primary targets for drug actions are as follows. First, they play an essential role in the biological life processes and pathophysiology. The structures of active sites of enzymes and the ligand binding pockets are highly amenable for high affinity interactions with small drug-like molecules. The presence of potential allosteric sites and the conformational variations in the binding sites. Also, they can be directly used as therapeutic agents. That is, agents for therapy. Enzyme structure and catalysis. Let us have a detailed look on the same. Enzymes are biological catalysts as well as they can act as receptors by acting with the substrates. These are the most efficient catalysts known in nature. They have the ability to enhance reaction rates by lowering the activation energy of reactions and also by stabilizing the reacting molecules at their activated complex sites. Stabilization theory of the activated complex by enzymes was first proposed by Linus Pauling. He concluded that the active site of enzyme is complementary to the structure of the activated complex so that the binding of the enzyme to the activated complex is extremely tight, which reduces the activation energy, enhancing the reaction rate. Next is the concept of enzyme inhibition. Although activation of enzymes may be exploited therapeutically, most effects are produced by enzyme inhibition. A survey reported in the year 2002 found that close to 30% of all the drugs which are in clinical use derive their therapeutic efficacy through enzyme inhibition, and then it was updated to include newly launched drugs, and it was found that 47% of all marketed small molecule drugs inhibit enzymes as their molecular targets. Any compound that slows down or blocks enzyme catalysis is termed as an enzyme inhibitor. This strategy can be exploited in correcting chemical deficiencies by inhibiting the enzyme which uses that chemical as its substrate. Like in seizures, which can be caused by insufficient gamma-aminobutyric acid, that is GABA levels. So thus, inhibition of the gamma-amino transferase enzyme results in the increased concentration of GABA, ultimately leading to production of anticonvulsant effects. Now students, here you can see we have depicted this figure. Chemical excess can also be corrected by inhibiting the enzyme that produces the molecule itself. Like gout results from excess uric acid concentration. Inhibition of enzyme xanthine oxidase, which converts xanthine to uric acid, which would result in the anti-hyperuricemic effects. Now again, this is depicted beautifully in the figure shown. Inhibition of biochemical pathways unique to a pathogen can reduce the growth or kill the pathogen, that is either bacteria, virus or a parasite. Like sulfa drugs act as a bacteriostatic effect by inhibiting dihydroterorate synthetase enzyme, which is required for the folic acid synthesis in bacteria. Humans do not require this enzyme as they can absorb folic acid directly from their diet. Here in this figure through the dihydroterorate synthetase enzyme. Now, here we have tabulated selected enzyme inhibitors which are used in clinics or they are in the clinical use. The compound acetazolamide has a target enzyme as carbonic anhydrase and it is clinically used to treat glaucoma, which is a vision disorder. Acyclovir, it is used for viral DNA polymerase and it is used in the clinical use for the treatment of herpes. If you look at allopurinol, it is using the enzyme or inhibiting the enzyme xanthine oxidase and it is used for the treatment of gout. Aspirin on the other hand, it inhibits cyclooxygenases and it treats diseases like inflammation, pain, fever, etc. Amoxicillin, it is inhibiting the enzymes which are penicillin binding proteins and it is used to treat a plethora of bacterial infections. Enalapril, this drug which is in clinical use, it is used for angiotensin converting enzyme and its inhibition and it reduces hypertension. Another is carbidopa, which is used to inhibit dopa decarboxylase and it is used in the treatment of Parkinson's disease. Digoxin, which contains or inhibits sodium potassium ATPase enzyme and it is used for the treatment of heart diseases. Lovastatin, this is used for the inhibition of HMG coenzyme A reductase for lowering of the cholesterol levels in the body. Methotrexate, that is used for the inhibition of dihydrofolate reductase and it is used for cancer and immunosuppression. Norfloxacin, it actually inhibits DNA gyrase and it is used very effectively in the treatment of urinary tract infections or popularly known as UTIs. Omeprazole, it inhibits hydrogen potassium ATPase and it is used to treat peptic ulcers. Viagra, it is used to inhibit phosphodiesterase and it is used for the treatment of erectile dysfunctions. Now let us study various types of enzyme inhibition. Enzyme inhibitors are molecules that interact in some way with the enzyme to prevent it from working in the normal manner. There are a variety of types of inhibitors including non-specific, irreversible, reversible, competitive and non-competitive bindings. Poisons and drugs are examples of enzyme inhibitors. Non-specific inhibitors. A non-specific inhibition affects all enzymes in the same way. Non-specific methods of inhibition include any physical or chemical changes which ultimately denature the protein portion of the enzyme and therefore irreversible. The process becomes irreversible. Temperature. Usually the reaction rates increases with temperature. But with enzyme reactions, a point is reached when the reaction rate decreases with increasing temperature. At high temperatures, the protein part of the enzyme begins to denature and thus inhibits the enzyme action. Now here students, you can see a flow chart showing different types of enzyme inhibitors. Acids and bases. Let us now talk about this topic. Enzyme activity is also controlled by pH. As the pH is decreased or increased, the nature of the various acid and amine groups on side chains is altered with resulting changes in the overall shape of the enzyme. Specific inhibitors. Let us talk about this topic now. Specific inhibitors exert their effects upon a single enzyme. Most of the poisons and drugs act by specifically acting on a single particular enzyme. What is reversible inhibition? Reversible inhibitors can bind to enzyme through weak non-covalent interactions such as ionic bonds, hydrophobic interactions and hydrogen bonds. Because reversible inhibitors do not form any chemical bonds or reactions with the enzyme, They are formed rapidly and can be easily removed. Thus, the enzyme and inhibitor complex is rapidly dissociated. And this can be of competitive type, non-competitive type and uncompetitive type. Let us now concentrate on competitive type of inhibition. A competitive inhibitor is any compound which closely resembles the chemical structure and molecular geometry of the substrate. The inhibitor competes for the same active site as the substrate molecule is competing for. The inhibitor may interact with the enzyme at the active site, but no reaction takes place. The inhibitor is basically stuck on the enzyme and prevents any substrate molecules from reacting with the enzyme henceforth. However, a competitive inhibition is usually reversible if sufficient substrate molecules are available to ultimately displace the inhibitor molecules completely. Therefore, the amount of enzyme inhibition depends upon the inhibitor concentration, substrate concentration and relative affinities of the inhibitor and substrate for the active site. Let us take an example to understand this topic in greater depth. Ethanol is metabolized in the body by oxidation to acetaldehyde, which is in turn further oxidized to acetic acid by aldehyde oxidase enzymes. Normally, the second reaction is rapid so that the acetaldehyde does not accumulate in the body. A drug which is called as disulfiram, that is antabuse, inhibits the aldehyde oxidase, which causes the accumulation of acetaldehyde with subsequent unpleasant side effects of nausea and vomiting. This drug is sometimes used to help people overcome the drinking habit. Second example, we take students of folic acid, folacin or pteroyl glutamic acid. Belongs to the group of water-soluble vitamins. Fresh leafy vegetables, green vegetables, cauliflowers, kidney and liver are rich sources of folic acid. The physiological function of folic acid coenzymes is in the synthesis of purine nucleotides and thymine, precursors in the synthesis of RNA and DNA intracellularly respectively. The folic acid coenzymes are specifically concerned with the biochemical reactions involving the transfer and utilization of the single carbon C1 moiety. Before functioning as a C1 carrier, folic acid must be reduced first to 7,8 dihydrofolic acid, H2 folate, and then to the tetrahydro compound H4 5,6,7,8 tetrahydrofolic acid, catalyzed by folic acid reductase, which uses NADPH as hydrogen donor. The participation of folic acid coenzymes in reaction leading to synthesis of purines and to thymine, the methylated pyrimidine of DNA, emphasizes the fundamental role of folic acid in the growth and replication of cells. Cancer cells grow more rapidly than the cells of normal tissues, and thus they have greater requirements for nucleotides as precursors of DNA and RNA synthesis. Consequently, cancer cells are generally more sensitive to inhibitors of nucleotide biosynthesis than are normal cells. Now here you can see the structure of methotrexate. Antifolates, folate analogues, aminopterin, which is 4-aminofolic acid, and methotrexate, amethopterin, 4-amino-10-methylfolic acid, are extremely potent competitive inhibitors of dihydrofolate reductase and thymidylate synthetase, and because of that this inhibits the synthesis of RNA and DNA. The application of methotrexate disturbs the metabolism of polyamines in rapidly growing tissues. Inhibition of polyamine oxidase, the key enzyme in the biodegradation pathway of spamine and spermidine, induced by methotrexate in regenerating rat liver tissue, is probably the consequence of inhibition of nucleic acid and protein synthesis. Now, after having studied the competitive inhibition, let us study a bit in detail about non-competitive inhibition. A non-competitive inhibitor is a substance that interacts with the enzyme but usually not at the active site, just like in competitive inhibition. The non-competitive inhibitor reacts either remote from very close to the active site. The net effect of a non-competitive inhibitor is to change the shape of the enzyme and thus in turn the shape of the active site, so that the substrate can no longer interact with the enzyme to give a reaction. Non-competitive inhibitors are usually reversible but are not influenced by concentrations of the substrate, as is the case for reversible competitive inhibitors. Uncompetitive inhibition is the third category of inhibition that we shall study. Those molecules that can only bind reversibly to an enzyme when the substrate is already bound to the active site are termed as uncompetitive inhibitors. In other words, the inhibitor binds to the enzyme substrate complex. Increasing the concentration of the substrate will not overcome the inhibition in this case. Indeed, the level of inhibition would depend on the sufficient substrate being present at the active site to make enzyme substrate complex. This inhibition is not very common, just like non-competitive and competitive. What is an irreversible form of inhibition? Irreversible inhibitors covalently bind to an enzyme because chemical changes to the active site of enzymes and they cannot be reversed. A main role of irreversible inhibitor students includes modifying key amino acid residues needed for the enzymatic activity. They often contain reactive functional groups such as aldehydes, alkenes or phenyl sulfonates. These electrophilic groups are able to react with amino acid side chains to form covalent adducts. The amino acid components are residues containing nucleophilic side chains such as hydroxyl or sulfahydryl groups such as amino acids, serine, cysteine, threonine or tyrosine. Irreversible inhibitors form strong covalent bonds with an enzyme. These inhibitors may act at, near or remote from the active site of the enzyme. Consequently, they may not be displaced by the addition of excess substrate. In any case, the basic structure of the enzyme is modified to the degree or the level that it ceases to work. Since many enzymes contain sulfahydryl alcoholic or acid groups as a part of their active sites, any chemical which can react with them acts as an irreversible inhibitor. Heavy metals such as silver, mercury, lead have strong affinities for the SH group too. Nerve gases such as DFP, which is diisopropyl fluorophosphate, inhibits the active site of acetylcholine esterase by reacting with the hydroxyl group of serine to make an ester. Oxalic acid and citric acid inhibit blood clotting by forming complexes with calcium ions necessary for the enzyme metalline activator. Now students, let us study the affinity labels or active site directed irreversible inhibition. Affinity labels are molecules basically that are structurally similar to the substrate for the enzymes that covalently modify active site residues. They contain a reactive functional group, so not only they can react with the active site of target enzyme, but also can react with many nucleophiles associated with many other enzymes and biomolecules. Consequently, these inhibitors display potential toxicity. Many cancer chemotherapy drugs are affinity labeling drugs or agents, so are not as common in drug design as other types of inhibitors. Let us take an example and understand this. Aspirin, as shown in the figure, the structure, irreversibly acetylates the cyclooxygenase enzyme, binding site preventing formation of prostaglandins. Binding to COX1, which is cyclooxygenase 1, inhibits its prostaglandin production by preventing catalysis of arachidonic acid, responsible for assisting platelet formation, thus causing the blood to thin and less the clotting process. Now here is the structure of aspirin. Another example, student, is that of tosyl 1-phenylalanine chloromethyl ketone, that is TPCK, which is a reactive analog of the normal substrate for the enzyme chymotrypsin. TPCK binds at the active site of chymotrypsin and modifies an essential histidine residue. Now what is suicide inhibition? These are modified substrates that provide the most specific means to modify an enzyme active site. The inhibitor binds to the enzyme as a substrate and is initially processed by the normal catalytic mechanism. The mechanism of catalysis then generates a chemically active intermediate that inactivates the enzyme through covalent modification. This process is called suicide inhibition because the enzyme is committing suicide by reacting with these drugs. The difference from affinity labels to suicide inhibition is that the requirement for an enzyme which reacts on it and converts it to a product which is actually an inactivator species. Let us better understand this with an example. Monoamine oxidase deaminates neurotransmitters such as dopamine and serotonin, lowering their levels in the brain. The drug salicyline, which is used to treat Parkinson's disease and depression, is a suicide inhibitor of monoamine oxidase. It binds selectively to monoamine oxidase B. Here you can see in the figure the structure of salicyline. Now students, after having studied such an informative module, let us summarize it. In this module, we have attempted to describe some of the features of enzyme structure and reaction pathways to make enzymes particularly attractive targets for drug discovery and design efforts. The essentials of drug discovery through enzyme inhibition have also been outlined in this module with the help of a few important examples.